All right. So we are going to be talking about sales for the next 4 to 5 weeks. Uh, uh, we are going to be taking a lot of notes. Sales is a huge unit that you will use not only in middle school, but you can also use in high school and in college. Um, and if you're going to be a nurse, doctor, anything in the science or medical field, you will have to know sales. Um, so make sure you grab a piece of paper. You better already have a pencil because y'all know we do worksheets every day or some type of something you have to write with. Um, so we are going to talk about the different scientists, cell theory for the next couple days. And then as you see in the background behind me, this is technically, if I'm not mistaken, it looks like it is a animal slash human cell. Um, and we will be talking about the nucleus, the plasma membrane, all that different good stuff, um, which we will be doing different activities. Some will involve candy, um, if we know how to act right. Um, but we'll be using lenses. We'll also be using microscopes because I have these slides that you'll see bacteria, different animal cells, human cells. Um, so cells were not even thought about. It took all the way up to the 1500s. This is during the Renaissance time period. You will learn in, I just had literally Mr. Vincent's class. I had to think about it. He will, I guess he will talk about that time period. Those are when the kings and queens were huge, the monarchies. And the one thing about the Renaissance, this is when people started looking into science, they were looking into education. And sales is one of those things that got discovered during that time period. Um, so, an oak tree shades you on a sunny day at the park. A rhinoceros wandering over to look at you at the zoo. After a rainy storm, mushroom sprouts in a damp woods. What do these things have in common? The oak tree, the rhinoceros, and the mushrooms. They are cells. They have cells within them. Um, so we will talk about eukaryotic cells and prokaryotic cells. Um, and you will determine which is which. In the back of the room, the cells are hanging on the wall um, with the different parts of them. Also, the eukaryotic and prokaryotic. What's the difference? So, when you are doing worksheets or lab work, make sure you use the posters in the back of the room. They will help you. Um, all cells are different shapes and sizes. They are not all the same. Um, they do different functions. As you see the bacteria cell, uh, that is the first one. And then you see the animal cell and also human cells also look like the one in the middle. And then the one on your right is a plant cell. Cells are the basic units of structure and function in living things. So write this down. This is something that you're definitely going to need to know. And also through your notes, there will be stuff highlighted in yellow. That means write it down. I forgot to do this slide. But the rest of them that are in yellow, only write that stuff down. And make sure you bullet point because not all of it is the same exact thing. So make sure you keep it separate. Microscopes. It is an instrument that makes small objects look larger. So we talked about lights in the last unit. And in microscopes, they have a convex lens. And this is why I told y'all to make sure you're paying attention because you never know when this stuff comes back up. So when you look inside of the microscope, the light from there will shine on through the lens, the light rays. And there's a mirror at the bottom, as you can see. Um, and you can see the reflection. And with convex lenses, it makes stuff that is small look bigger. So when you're looking at tiny amoebas or you're looking at different cells to determine what it is, or if you are, especially in the medical field, um, especially if you're a pathologist or you're a veterinarian, and most of the time, especially technicians, they will look at different samples to see what bacteria, and based on the shape and size, they can tell you what you have and what's going on. Um, and those jobs do pay really good money. Um, so we will be using the microscopes in this unit a lot so that you can see the different cells, especially plant and human cells, and also animal cells and probably some insects. Um, and then you will see bacteria. Uh, Robert Hooke. So he was a very interesting guy. So in the around the 1950s, I mean 1590, my bad, the invention of the microscope came around. And one of the people who were very instrumental in the microscope was Robert Hooke. Um, Not only microscopes, but also using them effectively to study how things work and what they look like. So he was born in 1635. He died in 1703. He became a scientist at 20, which a long time ago, you would pretty much, after middle school, technically, you would go to pretty much a college. Um, they skipped a lot of the stuff that we have now. Um, so that's why he was 20 and he became a scientist. Um, he was the one of the first people to observe cells. Um, he was also one of the people who observed the structure and the thin slices of different compounds through the microscope. He looked at the bark off trees, and that is where he started. And then he eventually worked his way up and looked at other things. Um, the one big thing about scientists is when they do research. And so when we were going over the scientific method, different parts of the research, you try to make sure that you keep accurate notes. Um, and when you feel like that you have came to the end of your research, then you publish. And so he published when he was 30 is Micrographia, um, which detailed different things about cells and his work. Um, he is first known for microorganisms in the form of fungi, which is fungus. Uh, he also paired with another scientist, Antonio van Leeuwenhoek, who discovered they both discovered working together, um, single cell life, and it took them 9 years to do it. Um, they used microscopes. He used microscopes. Robert Hooke did um, to look at different ancient cells. So anything that was fossils. He started off with wood, and then he looked at different other stuff. Um, because fossils can definitely tell. Um, and they also still have a lot of times pure DNA in them. He also became an architect later on in life after he was done being a scientist. He also worked on the other side and he worked with a little bit of physics. Um, he was mostly a biologist. Um, so you should have all those four things written down. Um, this is one of the microscopes he created. And as you see, there's an oil lamp and there's water involved. Like there's so much going on in this picture and different things, but this is how he was able to see those small negatives back then. Antonio van Leeuwenhoek. He was born in 1932. He died in 1723. Um, in 1974, he observed the first protozoa, and several years later, it was bacteria. Both of those are almost in the same family, and we will talk about them. He also began to observe tiny stuff. He was a Dutch businessman and he was an amateur scientist, which means he didn't really go to college to study and fully be invested like Robin Hook. So he kind of dibbled and dabbled. And being a businessman, especially in these time periods, they were all about making money. Um, and he made his own lenses to see in the microscope. He uses these lenses to contrast simple microscopes. So his microscope that he made, the simple microscope was in 1683. So 23 years later, after the compound microscope from Hooke, um, the modern day microscope hasn't changed since 1886. It there's a little different gadgets and there's stuff that a little bit better, but mainly it has stayed the modern compound light microscope, as it is called, has pretty much stayed the same since then. The reason why he found protozoa and bacteria was because he had a fascination of looking at pond water. And he was surprised to see one celled organisms, which he called them little animals because he didn't know the proper name. And he looked into different other species. He looked at the the plaque from your teeth. He would scrape it off and he would look at it, not only his but other people's, because he was a scientist and he's just fascinated with it. Um, he described for the first time, he looked at different sperms from insects, from dogs, from humans. Um, Stephen Helm was a co-discovery of him. He was pretty much like his partner also. Um, the difference between scientists then and now, they work together. Most scientists don't because they want the publication, they want to be that scientist who discovered. They also wanted the clout too, but they also work together. Um, he also studied the optical lens. He looked at the different, so he could see different things. Um, He was a very interesting character. Because he also looked at blood, red blood cells. Um, he contributed a lot to the cell theory. Um, he was a very interesting guy. To be a businessman, he did discover a lot.
Microbes are in us.
So make sure you put your notes up, because you will definitely need them. Um, make sure that you turn in your reflection worksheet if you have not got them yet. Hopefully, they are being passed out now. Um, answer the 6 questions. It will be an easy mastery grade. You just need to fill it out and answer the questions like you were supposed to. So, if you want an easy A, this is the easiest I can give you. I will also be looking for lenses because there's an activity that y'all are going to be doing using lenses. So hopefully y'all will be doing that tomorrow. Um, you will be finishing the rest of this slideshow tomorrow because you will be talking about cell theory. You will be also talking about the other scientists um, that you will need to know. And then we will be going over on Thursday and Friday the different parts of the cells. So, and we're gonna try to make it as easy as possible. So you will definitely need to keep your notes on that. Um, so if you don't have a folder or a journal, I would get one. I think Miss Coots sent that out on around the first week of school. But if you do not have one, you need to get one. If you think you're gonna lose it and need to keep it in the room, then either tell me or Mr. Ward and get something for you to put them in. But you will be doing an activity on Monday where you're going to need all these different parts of the cell because you are going to start labeling the different cells. Um, and we will walk through and we will do the basic animal human cell, and then we will go through and we will do the plant cell. They all are the same. The only difference is the plant cell has a cell wall and the animal cell does not. That's the only difference. Um, and then we probably label a bacteria cell. So this is what you will be doing with your lab partner that you will be getting. Um, and y'all will be working on this activity together and helping each other out. We will probably switch depending on your behavior. And if you are actually doing your work, probably switching, you'll get new ones. So don't get comfortable. And maybe be the person you want to work with, and maybe not, but you still need to do your work. Um, so turn your reflection sheet um, and make sure that you have a pencil and paper tomorrow.